Welcome to the Medical IsoTalks, your space for insightful conversations at the forefront of nuclear medicine and molecular imaging. I'm Dr. Mayara, nuclear medicine physician, and your host for today's episode. It's a pleasure to be here with you. Join me as co-host is Dr. Heber Videira, medical physicist and director of new business at Medical ALD. Today we have the honor of welcome a truly distinguished guest, Dr. Arman Rahmim is professor at the University of British Columbia and a distinguished scientist at BC Cancer in Vancouver. He's internationally recognized for his work in PET imaging, computational modeling, and molecular imaging therapy, with over 300 scientific publications and more than 200 invited lectures worldwide. Dr. Rahmim has received major awards from the Society of Nuclear Medicine and Molecular Imaging, highlighting his significant contributions to the field. In this episode, we'll explore an exciting and heavily evolving frontier: digital twins, PBPK models, virtual Theranostic trials, and the future of radiopharmaceuticals dosimetry. Let's dive into this fascinating discussion. Dr. Rahmim, welcome. It's a pleasure to have you with us. Professor Rahmim, today our episode will follow an interview format as we'll be learning about all of this from you, since these are topics that are not part of our daily practice in Brazil. So to start, Professor Rahmim, why is the traditional fixed dose model problematic?
Yeah, first of all, thank you very much for having me here. I'm very excited to be here. I appreciate your time. So to start, ba- basically the traditional fixed dose and fixed injected radioactivity, which is what we should really be more sort of accurately calling it, is basically the fact that historically in radiopharmaceutical therapies, which, as you know, are taking the field by a storm. They're being truly transformational, and there's a renaissance happening right now in nuclear medicine therapy. So historically, in 2001, for example, there was a publication where sort of started doing cycles of 200 millicuries being injected, and sort of from then on, people have sort of stuck with this 200 millicurie number. And so many clinical trials have happened. Approvals by in different entities have been based on this number, and it sort of has worked good enough to get approval. And of course, it's been making a difference in patient lives, extending life, for example. Having said that, it's quite problematic because it's not personalized by any means. Like, if you go to external beam radiation therapy world and you tell people, I don't know how much dose I'm giving you, people would be shocked by that, right? Like that's unacceptable in the external beam radiation therapy world, not to know how much dose you're giving to a patient. Well, that's what's happening right now. We don't know how much dose we're actually giving. We know how much injected radioactivity we're giving, but we don't know how much dose they're actually getting. And in those studies which have actually studied how much dose, how much absorbed dose is being done to the patients, you see significant variability in how much dose is being delivered to organs at risk and the tumor. So what this has resulted in is, because they don't want to, for example, be inducing toxicity in patients. So to be safe, we're kind of actually under treating many of our patients because we're not personalizing the doses that we're delivering to them.
That's perfect, Dr. Rahmim. And I would like to do one question for you. When we think about the limitation in real world practice, where does the variability must appear in the clinical practice? Tumor or organs at risk?
That's a great question. It actually appears in both, and there are studies showing that when you fix the injected radioactivity in radiopharmaceutical therapies, the organs at risk, such as the kidney and bone marrows, the range of doses that are absorbed could be one order of magnitude. So there's, for example, studies showing something like 18 times, 22 times between the minimum and maximum amount of dose that is being absorbed. And in tumors, it's even larger. In tumors, the variability could be something like two orders of magnitude, right? So for example, in a study I was looking at in dotatate, the tumor max compared to the tumor mean range between patients was something like 300 times different, and in PSMA like a factor of 30. So there's significant variability right now in terms of how much dose is being delivered to our patients.
So this naturally raises an important clinical concern. If we don't personalize therapy, what is the greater risk, loss or if loss of efficacy or increased toxicity?
I think it's actually both of them. I think both are happening. For example, in a good number of patients, probably most patients, we are under treating our patients. So even though the improvements in extension of life, for example, can be impressive, for example, 4 months, 5 months, even with this fixed injector radioactivity, you could imagine that the efficacy could increase and improve for a subset of patients that are being undertreated. At the same time, there is a subset of patients which might be getting, toxicity, which are getting toxicity, and we could actually tackle that as well by again not over treating and not under treating. So we could really play that game quite better if we're actually able to measure how much dose is being delivered to our patients, and to predict and anticipate how much dose they will get for different amounts of injected radioactivity that we give our patients.
Yes, perfect. Professor, to better understand when and potentially solve this variability, we often turn to modeling approaches. When we talk about the PBPK modeling, what are we essentially telling the history of the radiopharmaceutical inside of the body?
Yes, that's exactly what we're doing. So in there's two important terms for us to know, and I'll come back to the second term more elaborately later. There's pharmacokinetics, and then there's pharmacodynamics. So pharmacokinetics is what the body does to a drug, while pharmacodynamics is what the drug does to the body. So pharmacokinetics is really, as you say, it's telling the story of how the radiopharmaceutical moves and transports inside the body. And so PBPK models, which are physiologically based pharmacokinetic models, they're this sort of more holistic PK models that kind of link all the organs to each other, and they kind of provide a model of the human body. Of course, it's a simplified model, and there's a saying that says all models are wrong, but some are useful. It's very important to know that at the end of the day, no model ever fully captures the complexity and amazing elaborative details of the human body, but we're telling a story. We're coming up with models that are useful, and so then we could use those models for improved modeling of what will happen to the radiopharmaceutical once it's injected into the human body.
That's very interesting. And with that story, several biological parameters come into play. And among a receptor density, binding affinity, and the internalization, which parameters is most determinant?
So there's many different parameters in the body, in the PBPK models, and people do sensitivity analysis to really study, and there's mathematical tools for this. So because once you build this knowledge-driven PBPK physiologically based pharmacokinetic model, you could actually study which of these parameters have the biggest role. So we've seen in our studies, and we've sort of converged more on those which make the biggest difference. So for us, actually, receptor density is a big one. The flow, the vascular flow, is also a big one that that impacts the perfusion and penetration into tissue. Things like also release are important. So by release, I mean after something is internalized into the cells, then they can actually, there's a sort of a parameter that models how they leave the cells, and that ultimately does also impact things. So I think it's possible to narrow things down further to the most important parameters.
Yes, but Professor, one challenge is translating complexity theory into the clinical reality. How can models with more than 100 equations and 200 parameters be reduced to something applicable in the practice?
Yeah, this is a great question. I remember one of these grants that we were writing in Canada to get significant funds for these studies. Round after round after round, we were being rejected by our reviewers. And this was one of the key concerns that they had. They said just too many unknowns, too many equations, and how are you going to tackle this? So in systems biology, going back decades, people have similar problems, and there are solutions to deal with that, right? There's actually techniques out there, mathematical so-called model pruning or reduction techniques that are out there, sensitivity analysis that I just mentioned to you, to really study which of these parameters are more important. So there are advanced techniques that can reduce a very complicated model to simpler models. So in our own case too, we've kind of going from something like 100 or 200 unknowns, we're kind of down to, in our lab and in our developments, to something like 10 unknowns that we find are the most important ones, focusing really on the tumors, and also on a couple of organs at risk, like the salivary glands and kidneys, and a couple of key parameters within each of these, so that we could personalize those, and based on those, create digital twins for our patients.
Professor Arman, as these models evolve, they open the door to entirely new ways of testing therapies. So, what are virtual theranostic trials? What can they be used for?
Yeah. So yeah, I'm very happy to talk about this. So people have been talking about for a while now on so-called virtual clinical trials or virtual imaging trials, and this has been around for a while now. The idea has been historically, forget about the field of theranostics, more generally in the field of imaging, for example. The idea has been to, instead of starting with very, very expensive studies based on real patients, real, scanners, radiology interpretations, we simplify that, or start with virtual populations, virtual acquisitions, and even virtual interpretations. And the idea was to maybe use these to narrow down the highly multidimensional nature of all kinds of different parameters that we could be optimizing. For example, when you're doing imaging, you could be studying, when do I image? What is the duration of the scan that I do, right? What are the, for different metrics that you could use to assess the performance of your technique, right? Which kind of protocol do I use for image reconstruction? How many iterations? How many subsets? What kind of post-smoothing? So many different parameters you could play with. And of course, if you really do this in real life, and then want to show all those images, acquire a large population and show those to all those parameters to a real radiologist, that's just an extremely expensive sort of approach. But so to first start in the virtual world, that could make a big difference. People have also been talking about, both from different regulatory bodies, have been talking about in so-called in silico methods, or they call them sometimes computer modeling or non-clinical tests, to speed up approval of drugs. So I think these have been consistent with those. And in our sort of YouTube channel, we also have a couple of nice interviews with people talking about in silico imaging trials and things of the sort. So we've introduced the concept of extending those. We've introduced the concept of virtual theranostic trials, which sort of has the same spirit, that we're in the field of theranostics. We start with both PK and PD models, so pharmacokinetic models of how a radiopharmaceutical distributes in the body, and PD is the pharmacodynamics, is okay, what does that radiopharmaceutical actually do to the body, radiation biology and things of the sort. So we model those, so we were basically able to simulate radiopharmaceutical therapy in the virtual world, and then based on that we would generate a population, right, and then from that we could do a number of studies. So virtual theranostic trials, or VTTs in abbreviation, can be used to study many things. You could do protocol optimization, like what is that SPECT imaging, after you do therapy, in the course of therapy, that's best on this range of population of patients for dosimetry. What is the best pre-therapy PET scan protocol that allows you to better predict the future? What is the best way to digitally twin a patient? What is the best way or best amount of radioactivity to inject into a patient, personalized therapies? You could also use it to assess like AI techniques for lesion detection, segmentation, tracking, all kinds of things. And ultimately, once you run these virtual theranostic trials, you could actually gain the trust of clinicians, saying, listen, this is a very systematic study we did. We're really showing this is the more optimal way of doing things. And that can open them up to doing actual patient studies, more extensive and expensive patient studies.
Professor, I really agree with you that VTT is best tools to use if to answer a lot of questions in the development of our area, in principally the new radiopharmaceutical therapies. But I would like to discuss a little more one point, how do you build a reliable virtual patient population?
Yeah, so that's a great question. Ultimately, to do that, you need to have a realistic range of, for example, pharmacokinetic parameters, so that you create a realistic virtual population based on sampling of these range of pharmacokinetic parameters. So of course, you could look at the literature, because people have been fitting some of this data using different kinds of PK and PBPK models, and so people have been reporting. So there's a bit of a knowledge-driven approach, where you look at the literature, you look at the numbers people are reporting, and that allows you to put realistic numbers in there. And of course, the models themselves, as you build the different organs in the body and the compartments within the organs, right? So you have the vascular compartment, and then you enter the tissue, and then you bind to the cells, and then you internalize to the cells, and then you can leave the cells, all this stuff. So you sort of look at the literature, you look at the studies people have done, and you sort of create a knowledge-driven models that are also based on realistic parameters. You could also take these models and then further validate them based on your own data. So for example, we actually recently in our lab, a number of our trainees in our lab are really working on this heavily. For example, in our lab, James and TJ and Maziar have been really trying to fit the data that we have also to these models. And sometimes, for example, they would see that the data that we have, for example, in the salivary glands, doesn't fit really well to these models. And then we're like, why isn't this fitting well? Like, this should be working. And then we realize, oh, that's because we're missing one compartment that models non-specific versus specific binding of the radiopharmaceutical. So that allows us to go back to the literature, because the fitting is not working so well. And then we discover, oh, wait a minute, there's non-specific uptake that we weren't modeling. So let's add that in our model. Let's make our model more advanced and elaborate, and then go back and fit the data. So there's a bit of a back and forth between the with the literature that's out there, with the data that we're already having, and sort of trying to make these models more and more realistic.
Very interesting point, and everything comes down to trust in the results. And how can the simulations be validated to ensure clinical value?
Yeah, so I think that it's a great question, and these are really related to the answer I was basically giving right now, in that we look at the literature that's out there, real patient studies, and then we sort of are fitting those patients, and we're sort of getting a better sense and belief in our models that they're really capturing the truth well. Of course, the ultimate trust can be gained when, based on these simulations, we build, for example, predictive models or digital twins that are predicting the future for the patient, and we really basically show those predictions in blinded clinical trials to the clinicians and tell them, listen, this is what I'm predicting. How close are we to how things are? The truth is, it's easier than it seems, because right now we're doing things in common practice so poorly, in my opinion, that we can only do better. Right now, we're not doing much dosimetry. We're not measuring things. We're not predicting things. We're doing like the base way to do things, which is injecting the same thing to everybody. So right now, the uncertainty the uncertainty in prediction of what's going to happen to the patient is almost like 100%. Like the dose variability is like 100% variability. We think with some decent, and our evidence and others shows that with some decent modeling and machine learning techniques or digital twins, we can actually narrow down the predictions of the doses to patients, from 100% uncertainty to like 20% uncertainty. That's a step forward. So based on these models, then we can gradually gain trust. And running virtual theranostic trials themselves, doing it very systematically, thoughtfully, treating it just like it's real patient data, can actually open up dialogues with the clinicians, activate their imagination, and allow them to really use these tools more routinely.
Yes, perfect, Dr. Professor Arman. And daily, I work in the medical with medical physicists, and I produce some isotopes to do some research about the new radioisotopes, and we have copper 64, we have terbium evolution for the prostate cancer, and I would like to do one question for you. Can digital twins predict dose therapy in the PSMA before?
Yes, they can, and even more generally, even if we don't just focus on digital twins, even more generally, looking at machine learning techniques, there's more and more literature showing that we could narrow down that uncertainty in the predicted dose from, again, something like 100% variability to 20%, for example, right? So I think this is very important, and so there's more and more evidence being generated based on machine learning in general, computational techniques in general, and also digital twins more specifically, where you're actually building like pharmacokinetic models that are tuned to the individual patient to predict. So I think this is going to be a big deal. And one thing that came to me, and I meant to mention to you, is I really see the future where, right now we have oncologists on maybe 4 different fronts. We have medical oncologists, we have radiation oncologists, we have surgical oncologists, which are the surgeons, and we have nuclear oncologists. Well, I think, we've been talking with some of our great collaborators. We think there should be a 5th one being called a computational oncologist. And this person would almost be like an engineer sitting next to a pilot. This person would really have insights into the biological models, into AI, into digital twins, into sort of modeling combination therapies. And this person, just like an engineer sitting next to a pilot, would sort of work collaboratively to run these models and to advise the clinician who is making decisions for the patient that, I believe this kind of a therapy, this is the kind of a decision support that I can provide for you, that I make a recommendation that you do this kind of thing, because these models, including digital twins, are able to predict doses before therapy, for example, in PSMA.
Excellent. Professor Arman, and in practical terms, does this approach replace imaging-based dosimetry?
I don't think it will replace, dosimetry can be thought of two different things. One is retrospective dosimetry, which is when you make those measurements as they're happening. This is what normally dosimetry means. And then there's predictive dosimetry, which is what we're talking about. I don't think predictive dosimetry is going to replace retrospective dosimetry. It might reduce how much we do retrospective dosimetry. So, for example, instead of having to do many time points, it might reduce the number of time points that we need to do retrospective dosimetry. Also, even if I do a pre-therapy PET scan and I can predict how much dose this patient is going to get, we still have to do dosimetry to verify this was the case, and also to make further predictions for next cycles. So I think the future is going to be a mix of the two, where you do some retrospective dosimetry and you do some predictive dosimetry to allow you to better assess, number one, how the therapy is going, how much dose is being absorbed and delivered, and number two, what is, to personalize, how much injected radioactivities and at what intervals you need to be giving to the patient.
Perfect, Professor. Even within imaging, timing plays a critical role. Why does the imaging time window significantly impact dose prediction?
Great question. The key thing here, Dr. Heber, is the fact that these pharmacokinetic parameters that we want to measure and personalize for our patients, they can be measured with different amounts of certainty depending on how you image the patient. For example, some of these parameters, like the release rate, how things leave the cell, for example, you can really only start to see those effects later after injection. In the immediate, let's say after 1 hour of injection, you may not have had enough of a time to really observe how things are, for example, happening later in the process, right? So that makes it a big difference. Another big difference is how noisy our images are, right? So this also brings in, how good the scanner is, how sensitive the scanner is for different scanners of different sensitivity. We might need different kinds of imaging protocols to give us the sufficient ability to estimate the parameter reliably. So I mean, you mentioned also copper 64. So exactly. So in the past, people have been really focusing on fluorine-18, gallium, right? But I think with copper 64, because the half-life is longer, you could actually scan later. And then the ability, also with long axial field of view PET scanners, you can scan later reliably. So these open up the ability to scan later and therefore observe the sort of the late parameters, the pharmacokinetic parameters, more to estimate them more reliably, and therefore make better estimates of how much dose the patient will be getting in the therapy side, which, as you know, takes days. The therapy is taking days. And so what kind of imaging do we need pre-therapy to really predict what's going to happen in the course of days of therapy?
Very interesting point. Beyond the physics and the modeling, we also need to consider the biological response. And why is it important to model radiation biology in virtual Theranostic trials?
Yeah, this is a great question. And ultimately, we usually talk about predicting the dose, but really, our ultimate goal is not to predict the dose, but not just to predict the dose, but to predict how the patient will actually respond to therapy. Because that's all. So predicting the dose is just an intermediate step. So what we really, and as you know, it's not just about the physics; it's about the biology. And so the same dose, absorbed dose, if delivered with different dose rates in EBRT versus RPT, could make a big difference, right? So in EBRT versus in RPT, we're giving very different dose rates, and in RPT, because those rates are much smaller, the sort of responses would be very different. Many of these things people are not modeling accurately. Some of the dose limits, even like 23 gray to the kidneys based on the EBRT literature, those all need to be challenged. But the point is, ultimately, we need to be predicting response to therapy, and therefore we need to be running virtual therapies on our patients, and we need to be asking the question, what is the best way for me to treat this patient to have the best response and best treatment? And to do that, we need to couple the pharmacokinetics with the pharmacodynamics. And the pharmacodynamics for us is having very accurate, very reliable, very meaningful models of radiation biology.
And Professor Arman, and when optimizing therapies, how can we balance increased tumor dose and the organ protection when studying albumin binding?
So yeah, I mean, for example, albumin binding is something that people have, been designing new radiopharmaceutical new pharmaceuticals that would bind to albumin, which is this sort of protein that allows you to circulate in the body for longer and therefore stay there. And so what's very interesting is that using virtual models, using realistic models of PK, we can really understand, how much binding to the albumin can really change how patients get treated. This is something in real life is very, very hard to do. We could actually, in a virtual world, we could really simulate a very wide range of albumin binding and really understand very interesting phenomena that we were not even aware of by understanding these things. So again, so these kind of virtual models and these kind of pharmacokinetic models can open up new dimensions or new windows to dimensions of PBPK to RPT that we haven't been able to really understand before, just because it's such a variable and multidimensional world that you cannot fully study in the real world, just except with extremely expensive range of studies. So again, virtual therapies and virtual models and pharmacokinetic models allow you to really understand a lot of things phenomenologically and really gain insights. And based on that, you could actually design the next generation of radiopharmaceuticals, the next generation of radiopharmaceutical therapies.
Yeah, perfect, Professor. And in Brazil, I really like to see our science and to use it to put in the practice. But because I would like to do one question for you. What would be the first helix step for toward implementing these approaches in countries like in Brazil, for example?
Yeah. So thank you for that question. I'm excited to talk to you about this. I think Brazil is, for example, is a high middle income developing country, and there, it's even more broadly speaking, there's really fantastic science already happening in Brazil and in a number of developing countries. So I think what it takes to more specifically for the things that we're talking about, and more generally to do really fantastic science, is it starts with having a strong will by people such as yourselves, making the right partnerships, really understanding the strengths and the weaknesses and the opportunities and the threats that are there, and to make those decisions. So again, I mentioned sort of, there's different countries one can look at in the developing country world. China, which I'll come back to. China is a great example. India, for example, again, Brazil is truly shining in agriculture science and biofuels, right? So there's these sort of niche markets that have happened, and they're outpacing, science even in like America or Europe. In my own home country, Iran, like nanotechnology, stem cell research has been pretty outstanding. Turkey, civil engineering infrastructure. South Africa, actually RPTs, radiopharmaceutical therapies, and things like radioastronomy, et cetera, et cetera. So I think there's great science already happening in developing countries. Comes down to a strong will, making the right partnerships, and sometimes it starts, Dr. Heber, by mastery or even imitation of great science that's happening elsewhere, and then you sort of build on it. So the German philosopher Heidegger basically says, if you want to be inspired, first master what you're doing, become really skilled in what you're doing, and then inspiration comes to doing things that nobody else has ever done, right? So I think this is going to be very important. And again, as we look at this scientific sort of landscape, comparing places like America and Europe with other places, developing countries, many exciting things are happening. Politically, it's obvious. Like politically, I think developing countries are showing a lot more maturity and peace and need for security versus some of these countries. But scientifically, too, really great science is already happening, and I think there's fantastic opportunities.
Professor, thinking about the countries like Brazil, is it possible to start on a smaller scale without a large team?
Yeah, absolutely. I think a key point is that we live in a connected world, right? So we were just in Colombia for this World Federation of Nuclear Medicine and Biology, where I saw Dr. Heber as well, and a number of other colleagues from Brazil and other parts of the world. And so it was very exciting because we were sharing, the great things that are happening. But it's not just about learning from each other, but it's also about collaborating with each other. Now, perhaps partly due to COVID, it's far more possible to do virtual collaborations, and we kind of do that. Like in our own group, the digital twins and the computational nuclear oncology group that we have in our own group is actually not just in our group. We actively have weekly meetings with partners and colleagues from different parts of the world, right? So, making more investments in terms of staying longer with each other, making visits, having invited lectureships and things of the sort, planning, also benefiting from creative thinking. So again, in some of those countries, they have turned scarcity into an opportunity for innovation. Like in India and Iran, I'm very familiar that they take scarcity and take it into innovation. Advocacy is a big one that people sort of, even a small number of people can really advocate ambitiously to bring in more and more resources into what's happening. So going back to your question, in my career, Dr. Mayara, I've seen that a few people can sometimes make a huge difference, even in a center. So for example, I was at Johns Hopkins University for 13 years. I've seen many different sort of leaders and different entities and organizations. So it's become abundantly clear to me that a couple of key people in an institution, a couple of key people in a division can make a huge difference. And when they leave and another person comes and doesn't have that visionary, ambitious leadership, they won't have that. There's a really great book called Good to Great by Jim Collins. It's a sort of a classic in the business world. He talks about how leaders who are ambitious yet not egotistic and self-centered, so their ambition is not just for themselves but for the entity that they're leading and for their people and for their organization and for the well-being of the people around them, those kinds of people are some of the most impactful people. And again, with limited number of people, with ambition, with drive, with insight, with mastery, really great things can happen in this connected world that we live in.
Yes, great, Professor. And then finally, looking ahead to the future, what changes must we accept to enter the era of optimal imaging and personalized therapies?
Yeah, so great question. And again, I'll probably answer from a broader perspective of what do we need to do differently, for example, in different parts of the world, developing countries, etc., to be doing amazing science. I mentioned the example by Heidegger that it's important to be truly skilled, to master something before inspiration comes. So I've seen this, for example, in the example of China. I'll give you an example that, for a long time, people were saying, the Chinese tend to sort of copy or imitate the great science that is done. But the point is here, when you have drive, when you have investment, personal investment in terms of the amount of time and energy and effort you're putting in, good things can happen. And gradually you gain confidence, and then you start becoming very creative. So it's very interesting. In the past few years, I've made this observation, talked to other people too. I sit in conferences, and then I see some of the colleagues from, let's say, US industry versus Chinese industry, and I know them, they're my friends, and I see them and I talk to them, and I see this great confidence, for example, in the Chinese, right? And others, where now the US people, the US folks that I know, they're my friends, they're like, we're now doing certain things because these guys are doing these kind of certain things, right? Like United Imaging is a good example in our field. Like they're doing things that others are now saying, wait a minute, normally I wouldn't have done this, but they're doing it, so I'm going to do it. So there are a number of examples of this that are happening. So I think it's very important to think about these. It's of course important for us to have a mindset of a long term. This is a long term battle. This is about the long term sort of having a vision. And you see this in places like China and Singapore. Long term planning has made a big difference. Making creative industry academic alignment is very important. Like in South Korea, that's exactly what happened. Like a lot of strong industry academic alignment. So sort of reaching out, finding those entities. I'm going to end by a note. There's a very famous book and concept by Robert Greenleaf in 1977 on servant leadership. And he kind of made this discovery as he was lecturing and educating others that some of the greatest leaders that are out there are servant leaders. And he talks about this extensively. And so again, these are leaders who are looking for the well-being of others around them, for their people, for the people around them, for the people in their country, in the world, in the developing world, etc. Right? And so what they do is that these people, normally you don't even think of them as leaders necessarily. They're just there to serve. They're there to help other people. As you know, in medical physics, many people are passionate about helping others. It's a good thing that we tend to see. And so he says sometimes you don't even know these people are going to be the leaders, but they're the leaders of the future. They care, they're there to serve, they're there to help, they have empathy, and their ambition is for the well-being of others, right? So he says it's not clear that they're going to be the leaders, but they're the leaders of the future. And so I think we're waiting for those great leaderships and leaders of the future emerging from countries such as Brazil and many other countries in the developing world to really take a lead on many different frontiers, political, scientific, intellectual, all kinds of things. So I have high hopes of this happening. It does mean that we have to work hard. It does mean that we have to go back to our roots and to do soul searching and to talk to each other like we're doing here and to hope and aim for the best.
What a class did you have today? Professor Arman, thank you very much for sharing your insights with us today. And we deeply appreciate your time and your contributions to advancing the field. And to everyone who joined us, thank you for being part of this conversation. And if you enjoyed this episode and want to stay up to the date with the latest developments in nuclear medicine, don't forget to subscribe to the channel and follow us on our social media platforms, Medical ALD. We look forward to see you in the next episode of Medical IsoTalks. Bye bye.